Welcome learners, and today's topic is DNA binding domains. So in eukaryotes, genes are not simply expressed or not expressed. They are finely tuned to express various levels of gene product at specific times in specific cell types and in response to complex changes in the environment. To achieve this degree of fine tuning, eukaryotes take advantage of a wide range of mechanisms for altering the expression of genes. In contrast to prokaryotic gene regulation, which occurs primarily at the level of transcription initiation, regulation of gene expression in eukaryotes can occur at many different levels. These range from the initiation of transcription to the numerous mechanisms that modify mRNAs and their ultimate protein products. Several features of eukaryotic cells makes it possible for them to use more types of gene regulation than are possible in prokaryotic cells. Eukaryotic cells contain a much greater amount of DNA than do prokaryotic cells, and this DNA is complexed with histones and other proteins to form highly compact chromatin structures within an enclosed nucleus. Eukaryotic cells modify their structural organization in order to express their genes. So, regulation can occur at any stage in the expression of genetic material in eukaryotes. So, in this slide, you can see the various forms of regulation which is followed in as a molecular mechanism in gene regulation in the eukaryotes. So it starts with regulation of chromatin remodeling, regulation of transcription, regulation of splicing and processing, regulation of transport, degradation of mRNA, translational regulation and protein modification. Control of transcription by sequence-specific DNA binding proteins. Now, how does a cell determine which of its thousands of genes to transcribe? Is it based on a group of protein known as transcription regulators? These proteins recognize specific sequences of DNA, typically 5 to 10 nucleotides in length, that are often called cis-regulatory sequences. Transcription regulators bind to these sequences, which are dispersed throughout genomes, and this binding puts into motion a series of reactions that ultimately specify which genes are to be transcribed and at what rate. Transcription of each gene is in turn controlled by its own collection of cis-regulatory sequences. These typically lie near the gene, often in the intergenic region directly upstream from the transcription start point of the gene. Although a few genes are controlled by a single cis-regulatory sequence that is recognized by a single transcription regulator, the majority have complex arrangements of cis-regulatory sequences, each of which is recognized by a different transcription regulator. It is therefore the positions, identity, and arrangement of cis-regulatory genes which are important part of the information embedded in the genome that ultimately determine the time and place that each gene is transcribed. We begin our discussion by describing how transcription regulators recognize cis-regulatory sequences. So it was thought previously that these proteins might require direct access to the interior of the double helix to distinguish between one DNA sequences and another. It is now clear that the outside of the double helix is studded with DNA sequence information that transcription regulators recognize. The edge of each base pair presents a distinctive pattern of hydrogen bond donors, hydrogen bond acceptors, and hydrophobic patches in both the major and minor grooves. Because the major groove is wider and displays more molecular features than does the minor groove, nearly all transcription regulators make the majority of their contacts with the major groove. So molecular recognition in biology generally relies on an exact fit between the surface of two molecules. And the study of transcription regulators has provided some of the clearest examples of this principle. So in this figure, on the left, a single contact is shown between a transcription regulator and DNA. Such contacts allow the protein to read the DNA sequence. On the right, the complete set of contacts between a transcription regulator and its cis regulatory sequence is shown. The DNA binding portion of a protein is 60 amino acids long. Although the interactions in the major groove are the most important, the protein is also seen to contact both the minor groove and phosphates in the sugar phosphate DNA backbone. So transcription regulators, they contain structural motifs or domains that can read the DNA sequences. These are the four common types of DNA binding motifs. They are helix turn helix, zinc fingers, leucine zippers, and helix loop helix. In helix turn helix proteins, this is originally identified in bacterial transcription regulators, and this motif has since been found in many hundreds of DNA binding proteins from both eukaryotes and prokaryotes. It is constructed from two alpha helices. Here it is shown in blue and the red color, which are connected by a short extended chain of amino acid which constitutes the turn. The two helices are held at a fixed angle primarily through interactions between the two helices. The more C-terminal helix, which is represented in red color, is called the recognition helix because it fits into the major groove of DNA. Its amino acid side chains, which differ from protein to protein, play an important part in recognizing the specific DNA sequence to which the protein binds. All of the proteins shown here bind DNA as dimers in which the two copies of the recognition helix represented in red color are separated by exactly one turn of the DNA helix, that is 3.4 nm. Thus, both recognition helices of the dimer can fit into the major groove of DNA. The next one is leucine zipper proteins. So this motif is named because of the way the two alpha helices, one from each monomer, are joined together to form a short coiled coil. These proteins binds DNA as dimers where the two long alpha helices are held together by interactions between hydrophobic amino acid side chains that extend from one side of each helix. Often leucines are involved in this binding. Just beyond the dimerization interface, the two alpha helices separate from each other to form a Y-shaped structure which allows their side chains to contact the major groove of DNA. The next one is zinc finger proteins. This group of DNA binding motifs includes one or more zinc atoms as structural components. All such zinc coordinated DNA binding motifs are called zinc fingers, referring to their appearance in early schematic diagrams, which is shown in the left. It has a simple structure in which the zinc atom holds an alpha helix and a beta sheet together. This type of zinc finger is often found in clusters with the alpha helix of each finger contacting the major groove of the DNA, forming a nearly continuous stretch of alpha helices along that groove. In this way, a strong and specific DNA protein interaction is built up through a repeating basic structural unit. The next one is helix loop helix proteins. So related to the leucine zipper, the helix loop helix motif consists of a short alpha helix connected by a loop to a second longer alpha helix. The flexibility of the loop allows one helix to fold back and park against the other, thereby forming the dimerization surface. As shown in this picture, this two helix structure binds both to DNA and to two helix structure of a second protein to create either a homodimer or a heterodimer. Two alpha helices that extend from the dimerization interface make specific contacts with the major groove of DNA. Thank you all.